was the standardized cloning procedure that you guys had published on, which I think again kind of speaks to that point of if you're doing research and in comparing research, you need to have a standardized protocol, right?
Exactly. I mean, in this protocol, we decide to publish. I mean, we create that protocol was like an in-home. I mean, just for our lab, because we know as we're doing, all our studies, we have our all our mother plants in our lab. We have our mother plants room,
Hmm.
because all the studies we're conducting, we are taking the clones from one single mother plant. Because
Right.
actually, I don't know if you saw one of the studies that I sent to you. We couldn't get all the clones from just one single mother plant,
Mm.
and we ended up taking from two different mother plants from the same cultivar.
Okay.
And that the mother plant effect was bigger than anything else.
Huh.
So we thought that mother plant would not be an effect. We didn't even consider as effect in the beginning. But when we ran the statistics, mother plant, the two different plants, it was showing like a lot of differences. So then it comes to a problem of
So
cloning, labeling. Go
so
ahead.
were those mother plants both clones from an original plant, or were they grown from seed separately?
Clones from the same plant.
Wow, that's interesting.
Yes, that's and we found differences. And uh, so that's why we decide. Okay, right now, as we are conducting the next studies is gonna be all because we don't want to create another effect. So all the clones will be from just one mother plant. And as we create this cloning protocol to be, standardized throughout all our study, all the studies, and then as new students are coming, they already know what to do. It was like a extension publication. Then we decide to extend that, extension publication, and uh, um, publish. You know, because we have cloning or propagation protocols for other plants,
Hmm.
but not much for cannabis. We kind of like a hearsay. People
Right.
talk about good ways and good practice, and then we decide to publish that. Because again, that's another cool thing about working with cannabis. Because many things that we're doing is not crazy or very fancy or a rocket science, but it's something that was the first time done in cannabis.
Right.
So it's the same thing for the trigolinine study. Trigolinine it's an alkaloid, it's a plant secondary metabolite that is known as a water deficit stress marker for other plants,
Hmm.
but there is no studies addressing that in cannabis. And then I'm like, oh, well, let's be the first one to do it. And then we found that trigolinine is also a good way, it's a good marker for water deficit stress in cannabis.
Yeah.
So that's another another question that I guess was that I'm
Yeah,
kind of
no,
mixing
and
around.
and it's cool that you point that out that a lot of the things that you guys are doing, it's the first time it's ever been done in cannabis. And I think to your point too, perhaps it's taken for granted when you study other crops that there are standardized procedures for cloning, for measuring whether it's trichome density or anything like that. And the fact that you don't have that in cannabis yet makes it really hard to sort of compare results from other studies.
100%, 100%. Because some research that we're conducting in our lab, it's like it's being done, let's say 100 years ago for corn,
Right.
you know, but cannabis is a totally different plant. So maybe the responses are not the same. Of course, you can use that trend, you can use that information as a background, but it's not the same. It's a different plant,
Right.
you know. So that's another challenge that my students they have when they're writing like the introduction part of their like master thesis or PhD dissertation. They don't have much studies. They don't
Mm.
have much literature to include in their introduction. because there are not many papers. So the way they solve it is adding other plants, correlating
Yeah.
that approach with other plants.
Yeah. So we need more studies. We need
Yeah. So,
we
but we
need
need more funds.
we need more funding. Exactly. All right. Well, maybe that's a good segue.